This is a supplementary video for chapter 22 in the book called RNA Bioinformatics, edited by Dr. Picardi and published by Springer Nature in 2021. So iDEP web application for RNA-Seq data analysis. So the main thing we're talking about is a web tool called iDEP. This tool is a graphically with a graphical user interface and it's very intuitive. So the main thing about this chapter is they use a mouse data set treated with heat shock and with and without HSF1 knockout. So we first download this data from iDEP and prepare the data and walk you through step by step the analysis. The raw data is available here, and this is the iDEP paper that was originally published in 2018. iDEP is a rather comprehensive tool for analyzing RNA sequence data. Before you use iDEP, you need to summarize, you need to map these reads and obtain the reconst file or the FPKM or RPKM files. So what iDEP can only accept is a matrix of expression levels of different kind. After that, iDEP take it over from there and do gene ID conversion, data transformation, exploratory data analysis, differential gene expression, and enrichment analysis and pathway analysis, and a lot of data visualization. So we're going to follow the iDEP pipeline in this video and talk about these steps here today. This the URL for the tool is here, and you can contact us through this email. So first, let's talk about how we prepare data for iDEP. In this example, we show you how to obtain public data from within iDEP. So we go to iDEP website, and we click on the Analyze Public RNA-Seq dataset. This is a small app that enables users to download uniformly processed RNA sequence data for two projects, DE2 and ARC-HS4. These are efforts to massively reanalyze public RNA sequence data with the same reference genome and protocol. So we here for this example we choose the mouse data, and we say heat shock as our keywords, and we obtain these three data sets, and we're going to select the last one. And you can see here we have 24 samples in this experiment, and we're not going to use all of them. We're going to use probably just 8 of them containing just 2 of the genotypes and 2 treatments. So we're going to download the gene level data, open the gene level data in Excel. As you can see, this is the Excel, and I typically would like to see quickly how many genes are there. Well, here we have 32,545 genes, and we have 24 samples. Okay, 24 samples, and I mean, it's always helpful to take a quick look at this. And for example, we have a lot of genes, and for example, these guys, they are expressed almost 0 except this one across all samples. So these are mostly noise. So we need to remove these genes before further analysis. So we were better off with much less noise. Okay, so this will be our next step, even for these guys. So what we decided to do in the end is maybe at least one of the samples have decent measurable expression, at least a few counts, a few reads, okay, a few reads. For example, this will be borderline. I think we need, yeah. Because Excel sometimes can recognize gene symbols as text and convert them into date. For a date, for example, as EPT2 is a gene, it's a legitimate gene. But if you do that, well, here it's good. Sometimes it's converted into date. For example, here you convert it to September 2nd, it's a time. Okay, so make sure we don't we avoid that kind of thing. And to avoid that in that iDEP, what I did was I insert a space in front of this gene IDs, as you can see. So this will prevent auto conversion. So next, we'll take a quick look at the samples. So this is in iDEP, and this is intentional. You have the GSM number, accession number, followed by the sample title. So to take a better look, I think we could paste this, transpose this. Okay, this will give us a better view of the samples. There are 24 samples, and align this. We just need a subset. We just need we don't need the R6 R6. We don't need the HSHOC 90 related samples. So we are going to delete these guys from here. So we come back to here and we start deleting the HSHOC protein samples. So select all, make it a little bit wider, and delete the R6 numbers, R6 columns. So R6, 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 R6. Okay, so we don't not interested in here, and also the HSHOC protein. Okay, this is a HSHOC protein. So this, for example, this HSHOC protein, we're going to delete all of those. And we deleted R6 and HSP 90 related samples. So now we are good. We are left with 8 samples. And again, to take a look at this, we better copy this and transpose. Well, I'm going to use this again. So this is the original sample. I'm going to do it here, transpose. Okay. Now these are the 8 samples we have left. So as you can see, this is the mutant, this is the wild type, and we have control HSHOC, control HSHOC. So we could start to genotype. Okay. For genotype, we have HSF1, HIF1, okay, and wild type. This is rep 1, 2, 1, 2, 1. Okay, so we're trying to do here, we're trying to reduce the length of this long column name so that our figures come up easier. So my favorite way of typically of doing this is using Excel formulas. So to concatenate these 3 columns. So we have this and have a underscore, have this one, and concatenate another underscore, and the number of reps. Okay, so now you can see now we have this, and this together. Okay, and we can copy this. This works well even when you have more samples. Okay, for now, you can see, let's get rid of the formulas and with paste the samples. Now it's just sample names, and then we want to copy this back to the data file, so that okay, this is the original data file, and we're going to paste transposed, which paste transposed. So this will be lined up here. So this will show up in our figures. Okay, so we are ready to go, and we save this file. Next, we want to make a design file based on this. Okay, actually, this is the first. These 3 columns are contains all the information we want. So we will make a new file here, and we want to paste, transpose it again. So this is all we need for a design file. I think everything is here. So 2 factors, 8 samples. The column names has to match exactly. Since we copy pasted it from the same source, so they have to match. Okay, so we're going to save this as a design file. So make sure you save this as a CSV file, not an Excel file. So CSV file, okay, design, save, yep. I have a design file, and what do we do with this file, that intermediate file? I typically just save this just for future reference, as in case we mess up something. So I typically save this as an Excel file. So sample names, I just save this as a sample names file. As you can see now, our folder I have three files. The Excel file. This is the record of how we convert sample names. This is the design file. This is the original gene expression matrix, and we're ready to load this file to iDEP. So we come back, and I typically just leave these guys as default. This is readconst data, obviously, and we browse for the file, and this is the expression file, and this is the design file. After loading the files, you can see this is the design, this is the expression matrix, and the iDEP matched the most gene IDs to a mouse, and this is going to be used. Next, we look at the second step of the preprocess. So as you can see here, we uploaded the data to iDEP, and we go to the preprocess tab. As we talked before, there's several things that we need to do. The first is that there's a lot of genes that have almost zero or near zero counts that we need to get rid of. Secondly, we need to transform the data somehow, so that our gene expression measure is independent of library size or other artifact factors. Okay, so firstly, we'll take a look at these readconst data and total readconst. As you can see, most of them is around 20 million reads. So this is, and there's not a huge variation between, which is nice. And okay, and then in terms of the filtering, so right now we want to require genes have at least 0.5 counts per million and at least one sample. Okay, for 0.5 counts per million, and we have 20 million total for most of them. So that is to require that at least one sample have 0.5 times 20 is 10, have 10 reads. Okay, at least a gene to be retained should at least have 10 counts, 10 reads per sample. So this is fine, but it's rather conservative filter. As we can see from here, we only 13,848 genes pass the filter out of 32,000. So we probably we could benefit from just decreasing this filter. So we choose to 0.2 counts per million. This requires that a gene needs to be at least 4 reads in any of these 8 samples. Okay, so give us a little bit more samples, more genes. We have 15,000 out of the 32,000, and it's about 40%, 50%. I think that's about a good, very good number that I like to see typically. That's 40 for a given specific tissue. We only need a subset of the genes, so 40, 50, 60% is a very good number. I don't want to get rid of 80% of the genes, so only 20% left. Or I don't want to deal with 80% of the genes left. A lot of them is mostly noise. And look at is the scatter plot of this is the replicates, right? And right now the default transformation is the log to the count per million plus count, right? So the C is 4. So in other words, every number is added 4 and take a log 2. So this is the default, because it's pretty fast. And the most aggressive one, I think, is the RLog. And as you can see, once we switch to RLog, even the lower end, the spread is very small. So the R and the expression have a very close very close correlation. You can switch to this UN. As you can see, this will be different samples, right? So this is good, and also we can check the box plot, very nice, and also the density plot. There's a lot of other things that here that can enable you to interact with your data. For example, here is plot a gene. You can look at HSF1 itself, and you can see this wild type have higher expression, and the mutant have a lower expression. We could individual samples. It's not a huge difference. So in the mutant, it's still expressed. Maybe exome is I don't know how the knockout is done. Maybe they knock out a functional domain or something, but expression is still detected. You can search for the row numbers HSF1, for example, and this is the number, and HSF give us the other numbers. Okay, so interact with your data. I always recommend to interact with your data, to get some feeling about your data. It's almost like it is almost as important as keep going on, in dating and dates for a few years before you get married. So the get married part is the identification of differential gene expression and also the path analysis. Obviously, you can download the pre-processed data and converted cons data here. Step 3, we are going to look at the exploratory data analysis step. And in here, we're going to talk about heat map, k-means clustering, and principal component analysis. This is the tab that we are at right now. So by default, there's 1,000 tape. So the basic idea is that you take the 1,000 most variable genes, variable measured by standard deviation across all samples, and you show their expression profile. The reason that we only take the top 1,000 is because, again, a lot of genes doesn't respond to the experiment. Okay, so one thing that the first decision we have to make is that is 1,000 a appropriate number. So for some experiments like heat shock, radiation, these experiments are dramatic to the cell. So the cell will respond with thousands of genes, sometimes even half of the gene change expression. For some other experiment, it's very subtle. Sometimes you have only 100 of genes change their expression, sometimes even dozens, sometimes even none. Okay, and whatever the data tell us, it is what tells. I think people now trying to use gene expression profiling as a magic tool to measure anything. So one of the things that I did early in the days is to use cell phone radiation on a cell. The cell phone electromagnetic signal, that's very subtle. I mean, the number of genes changes is very minimal. So you want to switch that. One thing you can take a look is the gene standard deviation. So by default it's 1,000. So this is the vast majority of the genes, and this is some genes with extreme changes. Okay, but default is 1,000. To be safe, let's maybe move it to here, and we want to change into 2,000. So we want to change it to 2,000. We change it to 2,000, and this is how it looks like. And one thing we could do here is look at the tree with the sample name on it. This is very expected. We have the mutant here, the wild type here, and the replicates are grouped into together. You're starting to see that the mutant in one branch and wild type in branch. That means the genotypes have a bigger effect on the overall gene expression profile. A few other things that we can do to optimize these plots. The first thing is the color scheme. The color scheme by default is the green, black, and red, and this is nice to most people. But there are probably 10% of our population is colorblind, and so green and red is the most frequent problem, confuses colorblind people. I think there's a lot of uh, movement in this regard to to urge people to use the color scheme that are friendly to colorblind people. For example, you can change this. If you switch here, all the rest of the sample will change. Okay, so another thing that I want to switch here is that the cutoff z-score. So the color scheme, as we can see here, goes from -1 to 1 in here. Okay, and so sometimes this color range will be skewed by one gene showing a ton of expression. So we cut off with 4 standard deviation. In this case, I want to increase the contrast a little bit, so I decrease into 3. Here is the normalization. By default, it's just the center, but subtract the mean from each gene. And if you want to be more aggressive, you can further divide the standard deviation for each gene, and that is give you more information about about each of the genes. But that assumption has to be that each gene, all the genes show substantial change in their pattern, and you don't care about the relative magnitude of change. So typically, I don't use that. I still want our interest in the genes that they're relatively highly induced. For example, these guys. And then lastly, I want to change this to sample names, and this will change this color bar here. And as you can see now, we have 4 samples are labeled. Okay. Now let's take a look at this heat map and see what we can learn from it. So before we move ahead, I sometimes will change the width of the sample of the window, and then I can copy this to a PowerPoint and paste it here and start start discussing. As you can see from here, no, this one. You know, we can start to see the a group of genes starting to emerge. We can see the large group of genes here. This group of genes are highly expressed in the mutant, regardless of treatment. Okay, and on the opposite side, we have a large group of genes are highly expressed in the wild type, regardless of treatment. So these are the differences introduced by the mutation itself. Has nothing to do with the treatment. So and so that's why it's very important to delineate the effects of the treatment and the effect of the genotype. So that's why we need to use the interaction term later on to separate these different effects. And then you have this group of genes. This group of genes, as you can see, are highly expressed, highly regulated, upregulated by heat treatment in both genotypes. So these are the genes that are independent. These are the response to heat shock independent of HSF1, because they went up regardless of whether you have HSF1 or not. Okay. On the other hand, if you look at this group of genes, are only are drastically induced only in the wild type, which has the functional HSF1, but not in the mutant that lacks HSF1. So these are HSF1 dependent response to heat shock. The reason I spend so much time in here is that I want to show you guys that even before you start differential gene expression, you should starting to observe a lot of patterns and a lot of change. And this is also why this is also a time where you can observe the unexpected findings, and potentially if anything wrong with your experiment, you can start to see, replicas are messed up, and not grouped together, for example. That will be a red flag. So typically, I copy that and paste it in a PowerPoint and label that manually and start writing a discussion. This is as what I did in the so heat map and hierarchical clustering give us a very good overall bird's eye view of the overall change in gene expression. In k-means clustering, our goal is to divide the most variable genes into discrete clusters or groups, and then looking at the enrichment analysis, the enrichment for each of the groups. By default, we are taking down the 2000 genes. We discussed this in here in heat map. So 2000 genes is probably a good number. It captures the most variation. And into 4 clusters, you can start to see patterns. But the k equals 4 might not be the optimal for your data set. Typically, if you have many different groups in your data set, you will need a bigger number, or you have more samples, you need a bigger number. And you can use this as a guide. So the idea is that if you're looking for a number of clusters, that at which point a further increase in the number of clusters doesn't substantially decrease the total within group sum of squares. So obviously, if you stay at 2, obviously this won't be appropriate, because you can have one more and a substantial decrease in the sum of squares, right? But when it comes to here, this region is pretty murky. It's pretty very subjective. And it all depends on how granular you wanted the data set to be, and further discussions. In this case, we settled on 9. It's very arbitrary, actually. So we're going to change this into 9 clusters. Okay, with 9 cluster, we started to see a lot more details in terms of patterns of gene expression. In addition to large groups like this, for example, cluster, you have 336 genes. These are highly expressed in wild type. And we start to see a smaller group of genes where it is not very coherent within sample groups, but capture some random changes in here, group C. And you can take a look of these guys and start. You have genes that are group I genes, 101 genes that are responsive to heat treatment in both genotypes, and they have a smaller group I, group G genes, so only 24 genes. This group of genes are only responsive, and is induced by heat shock in the wild type animals, not in the mutant. Okay, so and then you could start to look at the enrichment result. There's 9 clusters. There's a lot of terms here enriched. So my suggestion is that it's focusing on the most significantly enriched term. For example, this one. Okay, focusing on the most and going and move down. And for gene ontology, go BP. There's over 6,000 terms. So a lot of these guys are, I think, is mostly noise. So don't try to make a story out of these guys by ignoring extremely significant result like this. So let's start from here. So the first one is fatty acid metabolic process. This is a surprise. I never know. I mean, I thought we were dealing with heat shock response, have nothing to do with lipid metabolism. And again, the cluster C is here. So this pattern is not correlated with our expression here. You'll see one, even within technical replicates, between technical replicates, there is a difference. But nevertheless, if you search the literature, you can find some evidence that HSF1 actually regulates cholesterol biosynthesis pathways, and is implicated in HCC treatment, in HCC hepatocellular carcinoma. And so this is often the case where you have one our understanding of their function of the gene is actually very limited. And you name it as H-shock protein 1, but you also have a certain. The second most significant category is these guys for cluster G, as we talked about. These are specifically induced by H-shock in the wild-type animals. As you can see, these are exactly the gene that we would expect. Response to heat, protein folding, and chaperone-mediated protein folding, and all that kind of stuff. We have 12 genes in this category. Not a lot, but it's still very significant. And to check further these genes, we can download the enrichment result and open it up. Once you open up, this is the detailed information, p-value or what, and also what genes are here. For example, if you go to group G, these are the group G, and these are the genes. As you can see, just by look at by the name, this is just mostly H-shock proteins. Okay, so the H-shock proteins, HSPA1, HSPA8, these are HSF1 downstream genes that are mostly clustered into cluster G. Okay, and then the third most significant group is cluster E, is enriched in circulatory system development, tube morphogenesis, and tube development kind of thing. This is development related. Again, this is unexpected. So let's look at the cluster E. Cluster E is this one, blue. This is actually very, very interesting kind of pattern. So this group of genes are highly expressed in the mutant without treatment. Okay, with treatment, this group of genes are downregulated. In the wild type, these genes are much lower expression, much lower, and is also lower and upon treatment. So this is interesting kind of group of genes, in the sense that it's specifically upregulated in this one sample out of 4. Now, does HSF1 is correlated with development? Well, if you look for the literature, what I did was just Google search HSF1 development. I ended up on this paper from the Morimoto lab in Northwestern, which I met him once or twice. And this paper is, I think, it's a review. And then what he said is that there's non-canonical rules of HSF1 and regulates development, reproduction, metabolism, and aging. Okay, so HS1 is a factor that actually implicated a lot of important biological and pathological processes, including cancer, as we see. But the danger in doing this kind of thing is the medical literature, the biomedical literature is vast, and there's a lot of them. It is is outdated, and a lot of them, frankly, is wrong. And if you have a vast database of publication that are very noisy, then there's likelihood that whatever you think, whatever your hypothesis, you're going to find some support in the literature. Okay. And for example, we have this paper, why most published research findings are false. I mean, so if we believe this, and then you have millions of papers published every year, and then whatever hypothesis you come up with, you're going to find support. So it's very important for us not to rationalize our finding. The last one that we want to and lastly, we can have a visualization of the enrichment result in this kind of tree. This is the innovation that we came up with in iDEP and ShinyGo. Try to summarize the relationship between different terms, and they're often very redundant. We cluster them by how similar they are in terms of the members. If they're similar, we cluster them together. And also this dot, the size of the dot represent the p-values. You can see these guys are important, are most significant. And then there's these guys, protein folding is most important in cluster G. And this is a very convenient way of overview. As you can see with the k-means cluster, we started to started to look into the different groups and the biological theme and the functions of the genes that are there. And also with this kind of exploratory analysis, we can look at the things that we are expecting for this group of genes. I'm not sure what is going on behind this, but the exploratory analysis could help us identify unexpected findings. And this will also form the foundation for differential gene expression and enrichment and pathway analysis. In principal component analysis, we want to produce a two-dimensional plot of the expression profiles so that we can investigate their relationship. For example, here it's the basic plot. Obviously, we want to, so the color coded for different genotype and the different markers coded for the treatment. Okay, as you can see, techno-replicates are grouped together, and these are the wild type before and after, and these are the mutant before and after treatment. We can also see that the first principal component captures 35% of variance, while the second component covers 29% of variance. If you want to go investigate further, you can switch to the third components. The third component carries 11% of variance. Typically, that's only reserved for larger data sets. Okay, for smaller data sets, I think the first two will do more than enough because it captures 64% of the variance. That's big enough. Now, the principal component analysis is a dimension reduction method, and so the component itself, strictly speaking, doesn't have any meaning. It's just a rotation of axes in high dimensional space. But for each component, for example, for the PC1, each sample have a projection, or a loading, have a loading on it, and this actually, so each sample will have a number. Because samples can divide into groups by these factors, we can obviously look at whether this principal component is correlated with any factor. In this case, iDEP automatically does that kind of analysis, analysis of variance for each of the factors. Didn't find anything, but in some other example, for example, in this one, the first principal component is correlated with the treatment, and even virally we can see the untreated ones is on the left side and treated ones on the right side. Obviously, this is the directional change of the gene expression profile there. But in this case, we didn't detect these kind of things. Another thing that we introduced in iDEP is that for each principal components, they have a rotation on each gene. So for each gene, there's a value. There's a value on each principal components. Okay, so this is a vector kind of thing. So you can treat them as a weight, weighted vector by each gene. So some genes have a bigger loading on this principal components. So in that regard, we could actually perform a sort of pathway analysis. Okay, this is the result. You can see principal component 1. So these gene ontology terms are enriched, and red means this pathway is more active, and for PC2, for the second and third principal components, the cell mobility and migration, so especially immune cell mobility, as we can see later on, would be more important. And on the other hand, principal component 5, these are suppressed. Okay, and principal component 4, fatty acid metabolism, is suppressed. These are innovative stuff that introduced in the iDEP, and for principal component 5, the immune system response is activated in principle 1, 2, 3, but it's a present 5. Other method you can try is a multi-dimensional scaling and Tsa and E. These are nonlinear dimension reduction methods, and for I think it's more reserved for larger samples. For this kind of sample, we don't really need. Obviously, you can download the coordinates of the rotation of the projection, and you can see so you can use other software to produce your own plots. So far here we conduct EDA on our expression data from different angles, from heat map, k-means, and principal component analysis. So these are very good tools that enables you to interact with the data, observe the data, and learn something with the data, some expected or unexpected things you can learn. And this is where you find the problem in your sample. This is where you starting to form the hypothesis. It's starting to form a intuitive understanding of your data, so which is very important, I think. So some people jump directly to different differential gene expression and pathway analysis. To me, I think that's skips a very important part of both the quality control perspective and the verification confirmation perspective. In the fourth step, we want to proceed to identify differentially expressed genes. So here is the this tab DEG1 and DEG2. Okay, we recommend user to upload recons data. That is because the recons returns more information, and also enables us to use the DSeq2 tool to more precisely model the distribution using negative binomial model. And the DSeq2 module is also more extensively tested. Okay, so here we want to select the factors and comparisons. Now we want to select factors and comparisons. We want to select these two factors, obviously, and we want the interaction term because that's the difference in response to treatment in wild type. So it's very important to set reference levels for different factors. Obviously, for treatment, control is a good one, but for genotype, we want to select, instead of using wild type, we want to select the mutant as the reference level. The reason being that the wild type had the functional HSF1, while the mutant doesn't. So this makes the interpretation of interaction term a lot easier. So this is what we're going to do. And also we want to select the direction of comparison. So for genotype, we're more interested in comparison wild type versus the mutant. So a positive fold change means a gene is highly expressed in the wild type. And the negative fold change will be the opposite. For treatment, we want to compare treated heat treatment versus the control. Okay, so a positive fold change will mean that these genes are upregulated by heat shock. And okay, we submit and recalculate. Okay, this is the results that we got back. And one thing, as I mentioned in the paper, so these numbers are a little bit on the smaller side. So I would like to decrease this into 1.5. By decreasing it to 1.5, we can see the number of genes that are up and down regulated right now. Okay, so there's a lot of information here. And that's because we have a models of genotype, treatment, and the interaction term. So some of the comparisons are telling us the effect of the genotype itself regardless of treatment. And then this is the treatment effect. And this is the differential treatment, the difference in response in the wild type animals. So to properly understand this, I think maybe this table will help a little bit. In essence, we have 4 biological samples. Okay, we have two genotype and two conditions, and then obviously a lot of questions could be asked. Okay, so for example, what is the what for wild type? What is the response with treatment? So this is the compare this sample against this sample. Okay, and the same question could be asked for the mutant. What's the response to hate versus the no treatment? Okay, and then obviously we can ask what's the difference in response. That precisely is the interaction term that we're looking for. On the other hand, we can also ask for questions like, without treatment in the control condition, what's the difference in baseline expression between wild type versus the mutant? As from the heat map, we see that there's a big difference there. The same thing could be asked about with heat treatment, what's the difference between the wild type and the mutant? Okay, so this is how we coded this. So wild type versus HSF1, wild type versus HSF1 under control conditions. So if since this is baseline, we didn't mention that, so we skip that. So wild type dash HSF1, that means the difference in this direction, and the same thing for HSF1, which is the reference level. The response to heat is heat versus control is in this direction. For other directions, for example, in here, wild type versus HSF1 for heat. So this means if we ignore these guys and just focusing on the with treatment, the difference between wild type and heat, so this will be the this factor. And then for the wild type, the response to heat for wild type is this. Okay, so that's how we interpret these guys, and you can see this interaction term. We have 111 upregulated genes. Okay, so the meaning for these guys are, so there's 111 genes are specifically induced by heat shock only in the wild type animal. Okay, so this is the other side. So these are the specifically downregulated by heat shock in the wild type animal, but not in the mutant. Okay, so this is exactly the most interesting one. Okay, so for example, and you can we talk about each of those and what it means. So heat versus control for wild type, and this is heat versus control in mutant. Mutant is not mentioned, and wild type versus heat shock in under control conditions, since control is a reference. Okay, we can download this gene list and take a look at. Here we see fold change. Here we see the genes and the fold change and p-value, fold change, p-value, and we even have raw data in here for your reference. Okay, so another thing we could do is to use the Venn diagram, and for example, we can take a look. For example, we can take a look at upregulated genes between the upregulated genes between the wild type, between the so this is the heat-induced genes between the mutant and wild type. So there's 186 genes in total. So from this number, as you can see, for the wild type, there's a lot more genes are being upregulated. Okay, so these guys are probably dependent on HSF1. Okay, now in the DG2 tab, we can look at each individual comparisons, as we call them, and to do some in-depth realization and analysis. Here, let's look at wild type, the response to heat in the wild type animals. You can see there's 511 upregulated by heat in wild type, and 219 are downregulated. Okay, very clear-cut response in this regard. And I mean, we can look at the genes that are highly expressed, highly induced. For example, this gene HSP1B is upregulated by over 100 times or 100-fold because this is log2. Okay, while this MMRN1 is downregulated dramatically. This is pretty interesting. The MMRN1 gene, why it is drastically downregulated upon heat shock. This is a massive soluble protein found in platelets and endothelium of blood vessels. Okay, so some kind of matrix protein. I don't know what the mechanism behind it to be drastically downregulated, but we could look at the scatter plot, for example. Okay, there's an error. I need to fix that error. But for the interactive ones, you can see this is HSPA1, this is HSPA1A, and SOX4, and these guys are highly expressed in. This is the heat treatment, I think, side. So there's a lot of these guys are being highly expressed. And then we can look at the gene ontology terms for downregulated genes is mostly development, cell proliferation, and the upregulated genes are more significant, as you can see. Okay, stimulus, tissue development, and then response to cell migration. There's a lot of other things in here. We can still look at this one to take a look. Response to stimuli development, and this is also tube development. For model organisms, iDEP also provide a lot of other different kind of databases. This includes microRNA and target genes, and also drug associations and transcription factor target genes. For example, here we can switch to a transcription factor target genes. Here we can see that among the upregulated genes, the most significant transcription factor target genes are H1F1A. Okay, so this is exactly as what would be expected. Okay, and for downregulated genes, to a lesser extent, but CMYC and other things. For the another factor is TP53, P53, and PPA gamma and PPA alpha gamma, and these things. So this suggests maybe TP53 is part of the response to the heat shock response. And there are actually papers regarding this. So these upon heat shock, so P53 protein is accumulated with heat shock. Okay, after looking at the wild type, let's look at the response in mutant. In the mutant, so this is in the mutant. Okay, in the mutant with the transcription factor, we still see TP53 here. So that means P53 is probably independent of HSF1. And if we switch back to we can see the downregulated ones are development. Upregulated are response to stress, differentiation, and tube. Going back to the transcription factor target genes, so we have RELA, which is part of NF-kappa B inflammatory response triggered by heat shock. Okay, and then lastly, we turn our attention to the interaction term. Okay, remember the interaction term captures the difference in response to the treatment. Okay, the difference in response to the treatment. For the transcription factor, we can see right now we have H1 HIF1A as a factor. The P53 one is gone, as you can see, and PPAR gamma and androgen receptor. And if you look at the genes, and these are HSP1B, HSPA1A, and SOX4. Yeah, it's a lot of the Hitchcock proteins, and if you switch back to Ontology, you'll see the upregulated genes that is related with protein coding, chapter-mediated protein folding, okay, response to stress, response to heat. So these are the HIF1 mediated response to heat stress. As we so for mouse, we have a lot of the databases here, and you can play with. So for example, here we want to look at the drug toxicogenomics data set, CDT. What we found here is that the upper downregulated genes are these genes overlap with certain drug-related gene set. Okay, as you can see here, we found a gadolinium is related, and if gadolinium is a compound that isolated from the Indian neem tree, was used to treat malaria, and it has some anti-cancer activities and also modulates Hitchcock protein 90. Okay, and this compound actually interact with Hitchcock protein, the Hitchcock process. Another thing that you can do at the DEG2 tab is to come here to use the string API. With the string API, we conduct independent analysis of the enrichment analysis for the upregulated genes is also protein folding. And another benefit of this is that we can enrich the protein domains. You can see there's Hitchcock protein 70 protein, and this is the name of the domain. Okay, is enriched, and you can also do the protein-protein interaction network here, which looks like this, and with a lot of links and interactions, a lot of links to literatures and annotation information. So this is on the StringDB website. What happens is iDEP sends the list of genes that you identified to the StringDB server, and the StringDB takes over from there. And lastly, for the comparison with wild type versus the mutant under control condition, so we're looking at what the mutation, the net effect of the mutation without treatment, and we see 473 genes downregulated and 302 genes upregulated. For the downregulated genes, you can see it's a lot of very significant terms related to metabolism, okay, metabolism development, and upregulated genes by the mutation. Upregulated genes in the wild type, these are highly expressed in wild type, include protein folding, and this is not very significant though. The fifth step, we are going to talk about pathway analysis. The difference between the enrichment analysis on the list of genes, we got pathways, and the difference here is that we are not limited by the list of selected genes. Rather, we're using all genes and to do statistics on it. So this result is independent of your cutoff for fold change and p-value. So that's the nice thing about it. That doesn't mean that the pathway analysis is superior than the enrichment analysis we did in step 4. To the contrary, so on step 4, the results are often less noise. Okay, you might lose something, but it's more have a better signal to noise ratio. For pathway analysis, often the power is not as good based on my experience. And also, iDEP have limitations in terms of the pathway analysis. We just use the full change numbers, and for some of the methods like original GSDA program, they can use they can use the expression data itself and do permutations and other things, which might be more powerful. One thing I'll stress here is that with tools like iDEP, it doesn't take much time. And also with so many methods, for example, we have so many methods here and so many databases, it's very easy to find something you're looking for. If you try different settings, it's very easy to find the story you're trying to create. So this is called p-hacking, or selective reporting should be avoided. This is number one thing that worries us as a developer of iDEP, in that it will be misused by people to tell the story they want to tell anyway. So we humans are typically very, very biased in terms of the things that we want to discuss and the evidence we want to see. So that's why I encourage people to verify and your result. Just report the robust results. A robust result will be something that could be fine using different methods under different settings and repetitively. Okay, so now something is so sensitive to a parameter that if you change it a little bit, it'll disappear. So this is in pathway analysis, we follow the same route. We're going to look at the heat shock response in wild type and the heat shock response in the mutant, and then we look at the interaction term, which is the difference of these two. So we heat shock in wild type with the default gauge methods. We didn't find any significant pathways. So gauge is set as default mostly because of its speed. So we turned on to the GSEA method, which is probably slower. This is slow because it conducts hundreds of thousands of permutations to evaluate the p-value. That's why sometimes the e-value, the adjusted p-value is the same. So here we are ranked. The pathways are ranked based on not but adjusted p-value, but based on the normalized enrichment score, as you can see from here. So the most important, the number one pathway is cellular response to heat and chapter-mediated protein folding. This is exactly what we're looking for in terms of HSF1 mediated HSF1 mediated response to heat shock. So this is consistent with the key means clustering and also the differentially expressed genes. For example, there's 54 genes in this pathway. Their expression is showed up here. As if you look at these genes, you see HSPA1B, HSPA1A, HSPA8, HSPD1. So these are, just looking at their name, you'll know that these guys are heat shock related. And obviously you can switch to other okay, so if you switch this to the transcript factor target gene database, and here we can see that our bibliography genes includes 37 HIF1A target genes. Okay, and we also see TP53 target genes as well. And these numbers will be much more than what we saw in DEG2 because we don't have cutoff anymore. I think in DEG2 there's 10 genes here. Okay, there's 10 genes there. That's the point of pathway analysis. Is, again, and you can see that these related factors we have HIF1A, P53, PPAR alpha, SPA3, estrogen receptor. Bear in mind that these p-values are not very significant. But you can look at the expression of these genes. It's not. Okay, next we switch this to a Cac database. Okay, switching to Cac, we found these pathways. Cac are typically very small, so you see a lot of familiar familiar pathways, but don't get too excited because it's very small, very easy for a pathway to show up in under Cac. But here the top upregulated pathway is protein processing in ER. And we can the pathway is automatically downloaded from Cac with the different players upregulated upregulated genes are showing red and downregulated genes in green. As you can see, there are a lot of upregulated genes here. And there are actually reports that heat stress activates ER stress signal, and because a lot of misfold proteins are accumulated in the ER. Okay. And the second contrast comparison that we're going to look at is the response in the to heat in the mutant. Without the HSF1, as you can see, the heat shock induces these processes. Look at them. It includes response to interleukin-1, and this obviously part of the inflammatory response here, and also a nice summary of this is probably this graph. As you can see, sort of two bigger clusters. Here is the response to cytokines and inflammatory response. These are mostly cell migration. Cell migration is mostly migration of immune cells. Okay, the upregulated genes. So these things are happening independent of HSF1. And there are papers seeing that histone factor 1 and suppresses the L1 beta through binds to the through interaction with the nuclear factor of interleukin-6. Okay, so finally we investigate the interaction term. Again, the interaction term measures the difference in response to heat. So literally, you can take the fold chance to calculate associated with the heat treatment in wild type, subtract that from the heat response in the mutant. Okay, so that is the difference in the fold changes, and will give us this result. Using GSE, and we can find that the upregulated gene, the most significant ones are chaperone-mediated protein folding, okay, response to heat, cellular response to heat, this kind of thing. The downregulated ones are regulation of macrophage migration, immune cell migration. So that means the functional HSS1, when with the heat shock, so this part of response observed in the mutant is suppressed. The PGSEA is a very nice visualization tool for pathway activities. We can see from here, we have all the four samples, four groups, A samples here, and this is the pathway activity. For example, the chaperone-mediated protein folding. So this pathway is only activated in wild type upon heat treatment. Okay, upon heat treatment, but cellular response to interleukin-1 is more highly expressed in here, chemokine pathways. So it's not much in here. So it's sort of the functional HSF1 is actually suppressed this response a little bit. On the other hand, the protein localization to centrosome is highly expressed in the mutant, not lower in the wild type. Okay, so this is a very nice pathway activity map that we can look at. Okay, finally, just put everything together, we can see that heat shock in mouse at least have HSF1 dependent pathways and also independent pathways here. On the left side, P53 seems to play a role. in mediating the stress response independent of HSF1, and have immune response, immune cell migration, also have an inflammation response. HSAC also induces inflammation response, and that response is somehow reduced by if we have a functional HSF1. And one last point I want to mention is a few things I want to mention is that we need to be critical of our results. In addition to other things, avoid p-hacking, try to use read counts instead of FTKM, and trying to find a robust result instead of the ones that you like that fits the story. You can make a story out of any gene or any data, and trying to use cross-validation. I tried to mention there are ways to validate your result. As you can see, the iDEP gives you opportunities to look into the pathways for many steps, starting as early as k-means clusters enrichment analysis, and the enrichment analysis of differentially expressed genes, and pathway analysis, and compare results of different pathway analysis methods, and use independent data sources and analysis methods. There's available in StringDB, also React on pathway analysis. These are all independent of iDEP. So if you can find your pathway through many times, that's a good indicator that this is something reliable, not some noise. So with the convenience of iDEP, we can produce a lot of results really quickly. So as the thing said, so iDEP actually gives you more power in analysis, but with great power comes with great responsibility, meaning that you can actually now make a lot of story out of your data. So I actually don't want that to happen. So I want you to be careful, be critical of your result. And there's so many reports out there that is not very robust. Thank you, and please let us know if you have any questions.